Alright, we got an interesting article just published yesterday. Rapid emergence of a maths gender gap in first grade. Do you even need any math in first grade? Anyway, here we report the results of a 4-year longitudinal assessment. Longitudinal assessment. What is longitudinal assessment? Of language and mathematical performance of all French first and second graders. 2,653,000 children, boys and girls, exhibited very similar math score upon school entry, but a gender gap in favor of boys became highly significant. After 4 months of schooling and reached an effect size of about 0.20 after 1 year. Whoa. Um, Although schooling will correlated with age, exploiting the near orthogonal variations indicated that the gender gap increased with schooling rather than with age. So these findings point to the first year of school as the time and place where maths gender gap emerges in favor of boys, thus helping focus the search for solutions and interventions. Alright, the data sites doesn't seem to be available right now. So let's look at another different thing. A lot of things has happened in 2001, as we all know. Also, this has happened: initial sequencing and analysis of the human genome. So this is the renowned popular paper article. Let's quickly look at the figure. Timeline of large-scale genomic analysis. Non-vertebrate model organisms in red. So top, it's like bacteria, etc. Then we have mouse. The blue ones are all mouse. Genetic maps, physical maps. So it started with around 1990. Some stuff started 1994. Then we have human genetic maps, physical maps, microsatellites, SNPs, ESTs, cDNA sequencing, full length, Spiral Project 15%, genomic sequencing, chromosome 22, chromosome 21. Alright, this is the idealized representation, hierarchical shotgun sequencing. So genomic DNA, from where they got the back library, organized, mapped large clone contigs, back to be sequenced, shotgun clones, shotgun sequence, SSCGT, from where assembly. Idealized representation of the hierarchical shotgun sequencing strategy. So this is the automated production line for sample preparation at the Whitehead Institute Center for Genome Research. The system consists of custom designed factory style conveyor belt robots. So this is more than 25 years ago, guys. They perform all functions from purifying DNA from bacterial cultures through setting up and purifying sequencing reactions. So finished and unfinished human sequence. Total sequence from the collection of sequence clones by sequence status. Number of clones, total clone length. What does it mean by total clone length? I want to know. Average number of sequence. So, in the context of genome sequencing, clone length refers to the average size of the DNA fragments. Okay. that are used as input for sequencing. So total human sequence deposited here, finished human sequence. As you can see, Whitehead Institute almost more than 1 million, finished 46,000. Then we have Washington University Genome Sequencing Center, Baylor College of Medicine, Stanford Genome Technology Center, Max Planck Institute for Molecular Genetics. So total human sequence deposited in GenBank by members of the Genome Sequencing Consortium as of October 8, 2000. So what are these maps? Let's take a look. What are these? Chromosome 2 map location, chromosome 2, chromosome 1, 3, 4, 5, 6, 7, 21, 22, X, Y, Jonathan map, gene map, Marshfield map, Yak map. Positions of markers on previous map. The key steps A to D in assembling individual sequenced clones into the draft genome sequence. A1, A2, then B, end-to-end alignment, okay. Alignment in middle only, not okay. A1F5 represent initial sequence contiguous derived from shotgun sequencing of clone A, and B1, B6 from clone B. Okay. Sequence clone A, sequence clone B, merge data, order and orient with mRNA, pair and reads other information, sequence quoting, contiguous scaffold. So here are the finished clones, chromosomes, all draft clones, pre-draft clones, sequence from contigs, sequence from clones. Interesting. 20 chromosome 21 and 22. There's nothing in draft clones. Clone level continuity, sequence level continuity, cumulative percentage. What does continuity means here? Contiguity or continuity. Proportion of the dump containing contigs of at most the indicated size. Okay, I have to look at something to understand. Okay, where does it stand today? So advancements in sequencing technologies, NGS, next generation sequencing like Illumina, enabling the simultaneous sequencing of millions of DNA fragments, providing a wealth of information, high accuracy sequencing, long read sequencing, whole genome sequencing, personalized medicine. Applications are here: early disease detection, complete human genome, filling in gaps and correcting errors from previous reference sequence, pan-genome projects, full genomes of diverse populations, further expanding our understanding of human genetic variation. Alright, so this is a very long article. We have only seen through 10, 11, past 10, 11 pages. It's like 62 pages. There is no need to UCSC draft human genome browser. publicly available sequence data, and other relevant genomic information. Histogram of GC content. So GC content is ratio of guanine and cytosine bases within a DNA or an RNA sequence. Significance of GC content can be correlated with various genomic features including gene density, replication time, recombination rates. Comparing GC content across different species can provide insightful evolutionary history and relationships. Can be an indicator of whether a gene has been acquired from a foreign genome. Can influence the design of PCR primers, and GC-rich sequences can impact primer binding and annealing temperature, thermal stability. GC pairs generally have higher stability than AT pairs. Oh, okay. So there is a lot of variation, like from 30% to all the way 60 70%. Okay. So it is not like it is constant. So other pair is of course ATGC, and um, in the case of RNA it is AU. In the case of DNA it is ATGC. For RNA AUGC. Length of chromosome arm and recombination rate. So shorter the arm, the combination rate is higher. Interesting. Average nucleotide substitution level for family. Number of kilobyte covered by family. So the average or median divergence of copies from the consensus is a particularly accurate measure of the age of the DNA transposon copies. So DNA transposons are DNA sequences that can move and integrate to different locations within the genome, so that they can go and combine in another place. Okay. More transposable. They are class 2 transposable elements, as opposed to class 1 retrotransposons that move through an RNA intermediate. Okay. Cut and paste transposable element mechanism of excision and insertion into target site.